Hey guys, it's Mr. Aragon. I'm here, obviously not here with you, but I'm here making this video for you guys. Hopefully clarify some things that you guys will be doing in the lab. The reason why it's so loud and echoey is I'm in a bathroom. It's the quietest spot that I could find in this place. All right, so what you guys are actually going to do today is you guys are going to finish your lab during 7th hour, and then you will finish your post-lab questions. And don't forget, you also have your mini research project that's due on Tuesday. Keep in mind, it shouldn't be meant to stress you out, and you only have to choose one of them. Answer the 7 questions for that particular part. And then create some type of creative representation for it. Okay. So what I want you guys to understand to help clarify some misconceptions or some problems that I've seen in the lab is you guys were given a lab like this, right? And what I want you guys to do is with me, I want you guys to actually fill this out, okay? So I'm going to zoom in here so you guys can see a little bit better. And in this lab, we're going to test for macromolecules, whether we have a lipid, a protein, a starch, or a simple sugar, right? So one of the first tests that you guys did, and it doesn't have to be in order, but one of the first ones that you guys did was a lipid test, all right? You were testing for lipids, and you're testing for the presence of lipids, right? You did this with a paper towel, right? The original color characteristics, so obviously if you chose milk, milk is going to be white, right? So you just write down the original color characteristic, right? The color that you're looking for, that's going to tell you if you have a positive for a lipid, you're looking for a clear color or see-through. There you go, see-through, right? The other test that you did with these 6 substances was the starch test. Right? And that is actually going to test for the presence of starch. Okay? I want to clarify one thing going back right here. Another name for the starch test is iodine test. Oops, iodine. Sorry, I'm typing with one hand. Or Lugol's test, right? And that's going to test for the presence of starch. So the original color characteristic, you're going to write that down for whatever substance. The color that's going to tell you, or the color that you're looking for to see if you have a positive for starch, it's going to be purple or black. Right? So the color that you're looking for to see if you have any starch, it's purple or black. So if you don't have a purple or black color, like if you have a green, a blue, or a yellow, that means it's negative. That means that substance does not have a starch. Remember, starches are complex sugars, right? The next test that we're going to do is the Benedict's test, right? And that's going to test for simple sugars. Right? The color that you're looking for to see if it's positive, it's going to be an orange or a brown. Right? An orange or a brown is going to give you a positive for a simple sugar. So if you have a green, a blue, a pink, or a yellow, obviously it's not going to be positive. It's going to be negative. The color that you're looking for is orange, right? The last test that you guys are going to do is the Biuret's test. Right? And that's going to test for the presence of protein. Right? And the color that you're looking for that is going to be a purple or a violet purple. Right? And you guys are doing these in the well, right? So it's a purple that you're looking for. So if the substance, after you've done the Benedict's test, has a purple color to it, then it's positive for protein. If it has a yellow, a green, or an orange color, it's negative, right? Keep in mind, when you do this test, the Benedict's test with the test tubes, do not fill the test tubes all the way up. You're just using a dropper full, right? So a full dropper, not a full test tube, full dropper. You squeeze it in there. Use the tongs that I've given you when you are removing your test tubes out of the hot water. Do not touch them with your bare hands, okay? Cool. So hopefully that clarifies the things that you're looking for. And then while you go, you're actually going to fill in this graph, right? So obviously, my first test that I did was a lipids test, seconds test. I have 2 carbohydrates test, Benedict's and then Lugol's or iodine, right? So remember, if I put Benedict's solution in water, I'm looking for an orange color, right? So if water turned blue, then it's going to be negative, right? Cool. And then once you're done with that, answer the post-lab questions, and then make sure you turn those in by Tuesday, right? And then we'll have a discussion pertinent to this on Tuesday. We're going to try and squeeze in another lab on how you can actually cook egg whites without using any heat. I know that sounds kind of weird, but we're going to cook egg whites without using any heat. Alrighty. So thank you guys so much. Please be awesome for Miss Nguyen. Remember that always wear your goggles and your apron when you're in the lab. And then once you guys are done, make sure you clean up all your materials. Keep in mind, none of the materials that you touch are very caustic or dangerous. But if you do get something on you, please wash your hands immediately, okay? Thank you guys so much. See you guys on Tuesday. If you guys have any questions, please feel free to email me.